And then we're happy to welcome Xintela and CEO Avi Lundgren-Åkerlund. Welcome, Avi.
Thank you, Cecilia. I will, during my presentation today, talk about progress in Xintela's projects, our exciting coming milestones, and also some background for you that may not be so familiar with our stem cell technology and our projects. Please note information in our forward-looking statements. So research and development is based on a specific cell surface marker, an integrin marker, which is called integrin alpha 10 beta 1. We have discovered this integrin on stem cells, and we use this integrin to identify, to select, and quality assure stem cells in the development of stem cell-based therapies. We have also discovered this integrin on certain aggressive cancer cells, and this has led to the development of antibody-based cancer therapies, and this is developed by our subsidiary Targinta. I will today focus on the stem cell-based projects. First, some highlights from my presentation. So we have developed a superior stem cell product platform, XSTEM, which is patent protected for all therapeutic use going forward. We have really strong preclinical data supporting the therapeutic effect of XSTEM. We have 2 projects in clinical development in osteoarthritis and difficult to heal leg ulcers. We've developed our own GMP manufacturing facility, so we produce our own stem cell products. And we can now also offer contract manufacturing of other advanced therapy products. Stem cells, mesenchymal stem cells, have a fantastic therapeutic potential. And that comes from their ability to regenerate and repair damaged tissues. They have the ability to develop into different cell types, like into cartilage cells, bone cells, fat cells, skin cells, and so on. So they can replace dead or damaged cells. They also secrete important factors like growth factors, immune modulatory, and anti-inflammatory factors. We isolate MSCs, mesenchymal stem cells, from adipose tissue. This is donated. We get our material from liposuction clinics, so there is no ethical issues here with this source. Then we have a unique technology to isolate MSCs. So we use our integrin marker to select and purify the stem cells. So we can avoid this problem with heterogeneous MSC preparations, and with antibodies and our marker we then can select out the MSCs to get homogeneous stem cell preparations, and that is XSTEM. And this has homogeneous preparation has many advantages compared to a heterogeneous. We can see functional advantages, better differentiation capacity, also improved secretion of some important factors, and also, of course, consistency or consistent high quality between donors and donors, because we get our materials from different donors and this heterogeneity will vary. So with our selection we get a consistent high quality every time. And as I mentioned, the product is also patent protected for all therapeutic use for a long time forward. We have our, which is also, this is really important, asset to Xintela to have its own GMP manufacture facility in-house. We have a license from May last year now to manufacture ATMPs, and with many great advantages, and among them reduce both production costs and risks of delays. So we will manufacture our own, of course, our own products developed in-house, and also our products developed together with partners and licensees, and also offer contract manufacturing of other ATMPs for clients. In our XSTEM pipeline, we focus on osteoarthritis and difficult to heal leg ulcers. So we have now entered into clinical development, and our clinical study in osteoarthritis has started in Australia. And we will start clinical studies on patients with difficult to heal leg ulcers after the summer. We also have prepared with great preclinical data a project in ARDS, acute respiratory distress syndrome, that we are ready to take into clinic when the time is right and when we have a partner on board for that. And we also evaluate, of course, other indications for XSTEM going forward. Osteoarthritis is the most common joint disease, where there is this progressive degradation of cartilage, joint breakdown, painful, debilitating, and we are many that suffer from this disease. And of course, there's a huge market. And we have no treatment. We have no treatment to offer these patients. Today, we treat symptoms, pain and inflammation, and many of us end up with having our joint replaced. And this is something we believe we can change with XSTEM-OA, that we have a safe and effective stem cell therapy for osteoarthritis. And this is really supported by our preclinical data, that we are developing a disease-modifying osteoarthritis drug that has the ability to stop further development of osteoarthritis and regenerate and heal the cartilage and reduce the need for joint replacement. XSTEM will be injected into the joint. This is an easy procedure for the patient and also an easy procedure for the doctor. We have shown both safety and efficacy of our integrin-selected MSCs in 2 horse models. We have shown that injection of MSCs into the ankle joint of horses with post-traumatic OA, and we compared this, so both joints had OA and one received the stem cells. And 6 months after the treatment, we could see great difference between the joints. This shows here, this is part of the data. Up here we have normal cartilage, the red is cartilage, and here is after 6 months degradation of the cartilage in the OA joint without treatment, and we could see significant less cartilage damage in the joint treated with osteoarthritis. We did a similar study where we actually looked at function, lameness. So here osteoarthritis was developed in these 2 groups of horses, and then one horse group received MSCs, and we could see that that significantly reduced the lameness, so hälta in Swedish, while the group that did not receive the stem cells, their lameness did not change. So we have been able to demonstrate both structural and functional treatment effects with our stem cells. We then wanted to look a little deeper into the mechanism of action of our stem cells. And we actually labeled XSTEM, our human product, with iron and with a red label, and injected these cells into a rabbit knee where we have a damage. Here with MRI you see the damage. 4 days after injection of our labeled stem cells, we could see that they were homing to the damaged. So here you can see this is filled up with black cells then containing the iron. When we took a deeper look into that tissue, we could see beautiful regeneration of the cartilage with our labeled cells all over this regenerated tissue. Here's the surface of the cartilage. Data really showing that XSTEM has a regenerative capacity. So these preclinical results that have been showing here now really support the therapeutic effect of XSTEM. So now we are of course very excited to evaluate XSTEM in clinical studies. So we have now started a clinical study in Australia on OA patients. This is a phase 2 1-2 study on up to 54 patients, and we are testing 3 different doses. It will be a single dose administered intra-articularly in the knee. We have safety and preliminary efficacy every 6 months up to 18 months, and we expect to have the first safety data in the end of 22 and early efficacy in 2023. The next clinical study for us is difficult to heal venous leg ulcers. They're called difficult to heal because they do not heal within 6 weeks. Very painful and debilitating for the patient and affect many, many patients. And here we do not have any treatment to offer these patients. We have also here evaluated XSTEM in a preclinical model, in a preclinical wound healing model. So we investigated the capacity of XSTEM to heal the wound, and we could see, this is the tissue in the healed wound, that the structure of the healed tissue is very similar to the normal skin. You can see very similar layers that you expect, when structure that you expect to see in the normal skin is also here, while the control wound without the cells had a poor healing. So with this data, we are now ready to start clinical studies, and that will also be a phase 1-2A study on up to 12 patients. A single dose will be administered topically onto the wound, and then after 10 weeks we will have a readout on both safety and efficacy. So this is a quite short study, and we expect to have results already in the end of this year. So we can conclude that XSTEM has really delivered the last year. We have granted patent for the XSTEM product for all therapeutic uses. We demonstrated safety and therapeutic effect and mechanism of action in 2 preclinical models for OA, in wound healing and also in ARDS. We have approval of our GMP facilities, so we can produce our own products. We have already successfully manufactured XSTEM for the 2 clinical studies, and started clinical study in Australia and submitted application to MPA, now waiting for the response so we can start the clinical study on venous leg ulcers after the summer. We also now have exciting clinical milestones to look forward to. So we're starting, as I said, the clinical study on difficult to heal venous leg ulcers after the summer, and expect then to have safety and efficacy results at the end of this year. And then also safety results from OA clinical study expected in the end of the year. Early efficacy signals from OA clinical study in 2023. So with positive results from our clinical study, we will have a great value increase of our projects, and that will also be a good time for enter into partnerships and license deals, which we expect can happen in 2023. We also plan to partner clinical development of XSTEM for ARDS and further development of stem cell treatment for animals at the current preclinical stage where we are now. With our great product, we have many opportunities to broaden XSTEM therapy to other indications, and that we will do when the time is right. We will be the exclusive manufacturer of XSTEM for partners and licensees, and the time is definitely right to offer contract manufacturing of ATMPs for clients. Before I end the presentation, I like to bring your attention to Targinta and the exciting work that Targinta is doing. So they're developing first-in-class antibodies targeting integrin alpha-10 beta-1 for treatment of aggressive cancers, including glioblastoma and triple-negative breast cancer. Targinta has selected 2 lead candidates and will soon initiate manufacturing of the antibodies. Directly after the summer, we plan to spin off Targinta, and that will then be followed by public listing. So shareholders in Xintela will receive equal number of shares in Targinta. You can read more about this and about information about Targinta's projects at Targinta's website, and also listen to presentations by Targinta CEO Per Norlén. Thank you very much.
Thank you, Evi. So speaking of Targinta, now that it will be spun off, how will the relationship look like between Xintela and Targinta going forward?
Well, Xintela and Targinta will be two separate companies. So Targinta will be independent and will be self-financed and so on. But of course we grew up together, and we have a lot to share. And also Per Norlén, CEO of Targinta, is the CMO of Xintela, and I am the CSO in Targinta. So we still have collaboration between the companies.
And then returning to a final question on Xintela, you have 2 clinical studies. You hope to have 2 clinical studies ongoing this year. So if we look ahead 1 year then, where do you see Xintela then?
Yes, in 2023, as I said during my presentation, we expect to have really exciting data from our 2 clinical studies, both from venous leg ulcers and from osteoarthritis. And with positive data from these studies, we have, in a perfect position to start negotiating with partners and to do license deals. And in 1 year from now, I also expect that we have, that our contract manufacturing is up and running, and that we are generating revenues from that.
Well, thank you so much, Evi.
Well, thank you very much.